I'm Keith Jerome. It's a pleasure to get to give the overview of defeatHIV and tell you what's going on in this second iteration of our collaboratory. From the very beginning, defeatHIV's really been built around a unifying hypothesis, and that has continued into this second iteration of the collaboratory, and that is that cell and gene therapy as a field really represents a uniquely promising approach to HIV cure. And I think that we're seeing more and more evidence of that in the meeting today, and we're greatly heartened by the number of collaboratories who have adopted these approaches into their cure efforts. In the first iteration of defeatHIV, we had a number of goals, but three that I'll really highlight here was we wanted to establish some robust non-human primate models in which we could test some of our potential interventions. We wanted to really maximize and optimize the disruption of CCR5, and then evaluate the immune control of the virus either after hematopoietic cell transplantation and in addition after transplantation with genetically protected cells. You're going to hear from Hans-Peter Kiem, my co-PI, in just a moment about more about the animal models in more depth, but we've utilized an SIV HIV HIV virus that among other characteristics includes the HIV envelope so that we can look at these CCR5-directed sort of therapies. We've established a very effective drug regimen, and I won't go into this in much detail in light of Hans Peter's upcoming talk, other than to say we can achieve robust suppression of virus, keep virus suppressed, and then once drug is removed, virus rebounds. And this gives us a wonderful situation in which we can evaluate some of these therapies. A number of findings from the kind of first iteration of our collaboratory, in which I'll just briefly highlight here. As I mentioned, we get suppression of viremia. The reservoir in these animals is established through a number of tissues throughout the body that very much recapitulates what we see in the human situation. We see T-cell depletion, the rebound of viremia, as I mentioned. Specifically, we see that these CCR5 modified stem cells mediate long-term multi-lineage engraftment. Hans-Peter will go into that more. We see selection of these cells after a SHIV challenge. And very relevant to the second iteration of our collaboratory, we see that this modification, this genetic protection of cells, actually has salutary effects to the overall immune control of the virus. So moving forward, we established this model, we established these findings, and we really, in thinking about what we would do in the second round, really wanted to say, looking globally at cell and gene therapy, what are the promising new ideas? Which of the cell and gene therapy approaches have unsolved roadblocks? Which ones look like they're promising to go forward? And we really identified three that we really thought would be most likely to make a substantial impact on the field over the next 5 years. Our collaboratory is built as a very highly interactive group centered around three projects right here in the colored boxes. They're supported by 5 scientific research supports or cores, as you can see here. And we have 4 industry partners, that's Juno Therapeutics, Gilead, Immunothon, and Sangamo Biosciences, all of whom interact with one or more of our initial research foci. The entire thing is then directed by input from our management and operations team as well as our community advisory board, and you'll hear more from Michael Luella about that in just a few minutes. So here's sort of the schematic that I want to use to present our 3 sort of lead concepts. And so the idea is here on the left, we have a reservoir of latently infected cells. Periodically, these reactivate and begin to produce virus. Those virions can then go on and infect new cells on the right, building up a cycle of infection here, or potentially reseeding the reservoir. And this is the process in which we need to intervene. So the first strategy that you'll hear some about in just a moment from Larry Corey is the use of chimeric antigen receptor T-cells. This project is led by Thor Wagner, David Rawlings, and Larry Corey, and the idea is to attack and kill these cells as they reactivate. So here in the middle, you can see the CAR T-cells in red. When the cells express some antigen, they're going to be attacked. And a corollary hypothesis, and something we can actually test here, is that the efficacy of this kind of approach might be maximized if it's combined with a latency reversing agent. Now, you heard this morning from Believe a competing hypothesis here, and I think it really illustrates the power of these non-human primate models to really test these competing hypotheses and understand how these approaches are actually going to work when they're combined. So we may get an anti-reservoir effect in the middle. We may also exert improved immune control of these ongoing cycles of replication with these CAR T cells. And you'll hear more about this from Larry in a moment. Our second strategy is the use of vectored or passive delivery of ECD4IG. You've probably heard from Mike Farzan in the past. These meetings or other places about ECD4IG. It's an incredibly potent and broad neutralizer of a wide variety of HIV isolates. And we would like to introduce this into our animals, either again as a passive administration or vectored through an adeno-associated virus with intramuscular delivery. And the basic idea is that if we do have an intervention that can achieve a reduction of the reservoir, for example, our CAR T cells, we may not get complete reduction of the reservoir, elimination of the reservoir. There may be late reactivations from rare remaining cells. A molecule like ECD4IG may be ideal to suppress the reactivation from those remaining cells. Now, that's one of the two hypotheses. The second point to really make strongly is that ECD4IG is actually a very potent mediator of ADCC. So not only may we get suppression of viral reactivation, we may actually get a direct anti-reservoir effect as well. Our third strategy is being led by Deb Fuller, Jim Mullins, and Hans-Peter Kiem, and Deb's here on the panel as well, who can answer questions about this as well as Hans-Peter. But essentially, if you think about where our initial two approaches, we've been attacking something in the process. This approach really seeks to protect the cells, the recipient cells over here on the right, and it's kind of depicted by this gray thicker bar that's saying these cells are somehow resistant. It might be CCR5 knockout, it could be introduction of a fusion inhibitor. And so our reactivating virus really doesn't have a place to go. But as I mentioned, we've seen that the act of protecting these cells actually has a beneficial effect on overall immune control. So these cells are protected, now they can mediate help, they can orchestrate a more effective immune response. And based on that, we hypothesized that the efficacy of this kind of approach might really be optimized if we can combine it with another way of revving up the immune response. If for this, we're using Jim Mullins' conserved elements vaccine that's been tested by a number of folks, and Deb as well. And so combining these may give us a very dramatic and powerful effect. So I don't want to go into this too much because Hans-Peter's going to talk a lot about the animals. But I will say that our animal studies are very, very tightly coordinated to make this as efficient as possible. So we have a single unified control group of animals that serves as the comparator for all of our interventions. Each intervention also serves as a comparator for the others. And this allows us to very quickly move from our initial studies of single interventions to identify the optimal ways that we can combine these together into what we hope will be a very effective way to suppress the virus. I'll have a couple of concluding thoughts. You can actually read these. I do believe in these combination of therapies, and I want to have something up here to read because I want to hand it over to my colleague, Hans-Peter Kiem, who will go into the next section of the talk.